What if we could rewrite the very code of life? Not just to fix a broken gene, but to improve on what nature gave us. To create crops that can survive any climate, or to eradicate diseases passed down through generations. This isn't science fiction anymore. This is CRISPR. And it's poised to change everything we know about biology, medicine, and even what it means to be human. To understand the future, you have to understand the past. For decades, scientists have dreamed of gene editing. We had clumsy tools, like the genetic equivalent of a sledgehammer. But then, we found a molecular scalpel hiding in plain sight, in the immune system of a bacterium. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a naturally occurring defense system in bacteria. It's their way of fighting off viruses. When a virus attacks, the bacterium captures a snippet of its DNA and stores it in its own genome, a kind of genetic, most wanted, poster. If that virus ever shows up again, the bacterium can instantly recognize it. Here's the genius part. The bacterium creates a guide RNA from that most wanted poster. This guide RNA acts like a GPS, leading a protein, called Cas9, directly to the viral DNA. Once there, the Cas9 protein, the molecular scissors of the operation, makes a precise cut, disabling the virus. In 2012, two brilliant scientists, Jennifer Doudna and Emmanuel Charpentier, realized we could reprogram this system. We could design our own guide RNA to target any gene we wanted, and Cas9 would go there and make a cut. It was a discovery so monumental it won him the Nobel Prize. This isn't just about cutting. Once that cut is made, the cell's natural repair mechanisms kick in. We can either let the cell fix the break on its own, which often disables the gene, or we can provide a new, healthy piece of DNA for the cell to use as a template. It's a find and replace function for the book of life. The most immediate and promising application of CRISPR is in medicine. Think about the thousands of diseases caused by a single genetic mutation, diseases like sickle cell anemia, cystic fibrosis, and Huntington's disease. For decades, these have been life sentences. But with CRISPR, we can now correct the underlying genetic error. Take sickle cell anemia. The disease is caused by a single mutation in the beta-globin gene, causing red blood cells to become a rigid, sickle shape. This leads to pain, organ damage, and a shortened life. A recent CRISPR-based therapy, Casgevy, was the first ever CRISPR treatment approved for use in humans. It works by taking a patient's own stem cells, editing them with CRISPR to turn on a different gene that produces a form of hemoglobin that doesn't sickle, and then re-infusing them. It's a one-time treatment that's essentially a cure. And it's not just monogenic diseases. Researchers are using CRISPR to fight cancer by engineering a patient's T cells to better recognize and destroy cancer cells. They're developing therapies for HIV by snipping the virus's DNA out of infected cells. The potential is enormous, and clinical trials are happening all over the world. But here's where the conversation gets a lot more complex. The therapies I just mentioned are called somatic cell editing. They only affect the patient being treated. The genetic changes are not passed down to their children. But what about editing the germline, the DNA of sperm, eggs, or embryos? Germline editing would allow us to prevent inherited diseases from ever being passed on. Imagine a family plagued by Huntington's disease for generations, finally able to have a child who is guaranteed to never get it. The therapeutic benefit is clear and compelling. However, once we cross that line, a fundamental question emerges: where does therapy end and enhancement begin? What if we could edit genes to not just prevent disease, but to make someone taller, stronger, or smarter? To give them perfect vision or an extraordinary memory? The technology that could cure a genetic illness could also be used to create a designer baby. This isn't a futuristic fantasy. This is a very real, and very heated, ethical debate happening right now. The concerns are immense. Who decides what constitutes an improvement? Will these enhancements only be available to the wealthy, creating a new class divide of the genetically haves and have-nots? What are the long-term, unforeseen consequences of making permanent changes to the human gene pool? The scientific community has largely agreed to a temporary moratorium on germline editing, but that hasn't stopped some from pushing the boundaries. The infamous case of the Chinese scientist who created the world's first CRISPR-edited babies in 2018 served as a stark warning. He claimed to have given the twins a mutation that made him resistant to HIV, but the experiment was widely condemned as reckless and unethical. It demonstrated how easy it is for scientific ambition to outrun ethical responsibility. But CRISPR isn't just for humans. Its true, unexpected future might be in our food, our environment, and our fight against climate change. In agriculture, CRISPR is a game changer. We're not just creating GMOs in the old, controversial way, by introducing foreign DNA. Instead, we're making precise, surgical edits to existing genes. Scientists can create crops that are resistant to drought, disease, and pests. They can make them more nutritious or extend their shelf life. We could develop climate-resilient crops that can feed a growing global population in an increasingly unpredictable world. Take the humble mushroom. Using CRISPR, scientists have edited a single gene to prevent it from browning. The result is a mushroom that stays fresh and white for longer, reducing food waste. This isn't science fiction, it's already here. CRISPR also holds the key to fighting invasive species and disease-carrying pests. A technology called a gene drive uses CRISPR to ensure a specific gene, and any modification made to it, is inherited by every single offspring. Imagine releasing mosquitoes carrying a gene drive that makes them resistant to carrying malaria. Within a few generations, you could wipe out the disease entirely. Or consider the possibility of using CRISPR to bring back extinct species. The idea of de-extinction isn't a joke anymore. Scientists are working to bring back the woolly mammoth by editing the genes of an Asian elephant. This could restore lost ecosystems and help fight climate change by revitalizing the Arctic tundra. But it also raises ethical red flags. Should we be playing God with nature, and what happens if we get it wrong? CRISPR is an incredibly powerful tool, but it's not without its challenges. There are still technical hurdles to overcome, like ensuring the cuts are always precise and don't cause unintended, off-target, edits. There's also the question of delivery. How do we get the CRISPR machinery to the right cells in the body without causing harm? Researchers are working on new and better versions of CRISPR, like prime editing and base editing, that promise even greater precision and control. Ultimately, CRISPR is a reflection of ourselves. It's a tool that can be used for immense good or for catastrophic harm. The future of this technology isn't just a scientific question, it's a societal one. We have to decide, as a global community, how we will use this unprecedented power. We have the ability to cure the sick, feed the hungry, and protect our planet. But we must also decide where to draw the line. The story of CRISPR is still being written. And we're all a part of it.